Hi everybody, this is your TF Mary, and this is a supplemental video on protein structure and how it changes during cooking. Not all of these words in the title will be familiar right now. Denaturation is just another word for protein unfolding. And coagulation is what happens when proteins come together to make food solid, like the ricotta cheese that you're going to make in lab in the next week or two. You'll learn all these concepts a little bit better as you watch the video. So what you're looking at here is a close-up view of a protein that has been unfolded and stretched out as far as it would go. You learned in class that a protein is a polymer made up of amino acids, and each amino acid here starts with a little blue nitrogen and ends with a carbon double bound to a red oxygen atom. So each of these amino acids has a backbone that is exactly the same as the others, but each amino acid also has its own side chain that has a different shape and different charge and polarity. So let's take a closer look at a few of those. So let's start with this amino acid over here on the left. It's called lysine. And one thing you're going to notice as you compare that to the structure here on the right is that none of the hydrogen atoms are shown. When hydrogen atoms are shown in structures, they're normally white in color. In proteins, we normally don't show them because they take up so much space and make it so difficult to see what else is going on. This amino acid has a positive charge, which you can see indicated down here. That would allow it to participate in ionic bonds, which are a very strong type of noncovalent bond. We'll talk about it a little bit later. So next let's take a look at this amino acid called phenylalanine. If you take a look at the side chain, you can see that it only has carbons and hydrogens in it, and that makes it very nonpolar. So this is an amino acid side chain that would like to be away from the water, which is what the protein is floating in. That means that when the protein folds up, this amino acid will probably wind up on the inside. Okay, so now I've zoomed out so you have a bird's eye view of the whole protein. For reference, earlier we were looking just at this lysine and phenylalanine here. The typical protein has around 300 amino acids in it, and as you can see they're all linked together through one long chain. There is one additional covalent bond right here. And now if I zoom in on that spot, you can see that that extra bond is formed between the sulfur atoms on two cysteine amino acid side chains. So this is an interesting type of covalent bond in that if you lower the pH, you can actually break this bond, whereas normally a covalent bond is very stable. Okay, so now we're taking a look at the protein in its folded state. Instead of being stretched out end to end like you saw earlier, now that long amino acid chain is curled up on itself. And if you're like me, you look at this and you can't figure out where one end is or the other, and it's hard to tell what's actually going on here. So what I'm going to do is show you where that backbone is, that long thread that connects all of the amino acids together. In this representation, you can start to see that the protein is doing a lot of interesting things. That long chain of amino acids is sort of curling in a helix, or it's going back and forth in strands. But it's still very difficult to see what's going on because of all of those side chains, so let's hide those as well. Okay, so now we can finally start to see where this protein begins and where it ends. If you gave me a piece of string and asked me to fold it up like this protein, I would have a really hard time. But somehow, inside of a cell, the protein folds up correctly every time. And that's because it's stabilized by these extra non-covalent bonds that help it retain its shape once it folds correctly. One of the major contributors to the protein's stability is hydrogen bonding. And when you see in a cartoon representation either arrows that look like this, or helices that look like this, what that's trying to tell you is that there are a lot of hydrogen bonds in a very particular orientation there. And in order to see what those look like, we're going to have to switch back to the stick model. Okay, so that got visually complicated pretty fast, right? It's difficult again to see what's going on. You learned earlier that these amino acid side chains do a lot of interesting chemistry, but here the hydrogen bonding is actually between the atoms in the protein backbone. So I'm going to hide the side chains again. So now we can finally start to see something that looks like that curlicue structure you saw in a cartoon earlier. So remember, there are no hydrogen atoms shown here, but each of these nitrogens will have a hydrogen atom pointing upwards, and that hydrogen atom is forming a hydrogen bond with the oxygen above it. There were actually a lot of hydrogen bonds there, and remember, this is only one small portion of the protein. In order for the protein to unfold, each of these hydrogen bonds would have to break, which is energetically costly, but then since the protein is floating around in water, new hydrogen bonds will just form with the water. So this doesn't create a really big energy difference to help the protein stay folded. We need to look at some other bond types, like ionic bonds. So here's our eagle-eye view of the protein again, and this time I'm going to zoom in on this part of the picture where there is an ionic bond. I had to hide a few amino acid side chains to make this more visible. but the bond that we care about is between this oxygen, which is on an amino acid called glutamate, and a nitrogen on this amino acid called arginine. Now that probably doesn't look very different from the hydrogen bonds you saw earlier, but if we look back at the structure, we can see that actually each of these atoms is charged, and they have opposite charges. So this must be an ionic bond. This is a very strong type of non-covalent bond. There's another major force that helps the protein stay folded called the hydrophobic effect. This protein would be floating around in water, typically inside of food, and the amino acids that have side chains pointing towards the outside of the protein are mostly hydrophilic. That means they like to interact with water. But the side chains that point towards the center are mostly hydrophobic. They can't form hydrogen bonds with water, so it's energetically favorable for them to be stuck in the center of the protein near each other. So I guess the take-home message for this part of the talk is that once the protein folds up, there are hundreds and hundreds of interactions like hydrogen bonds, ionic bonds, nonpolar interactions that keep the protein folded that way. So now let's talk about how we cause proteins to unfold during cooking. One of the ways that we cook foods is by raising the temperature. I've drawn here two amino acids that are involved in an ionic bond, but the same sort of process would happen for a hydrophobic interaction or a hydrogen bond as well. On paper, these amino acids don't look like they're moving very much, but in reality, each of these atoms would be moving around a little bit, and as we raise the temperature, that motion increases as well, and eventually that will tend to break this bond. After the bond is broken, the amino acids can float away from each other, and the protein then unfolds. So another way we can break the ionic bonds, besides raising the temperature, is by lowering the pH. It turns out there's a reaction between the protons that we add when we lower the pH, and the negatively charged side chains on amino acids like glutamate and aspartate that participate in ionic bonds. And that reaction results in a neutralization of the charge. Once it's neutralized, that amino acid can't participate in an ionic bond anymore. And the more of these protons we add, the more we push the reaction over to the right and get more things that look like this. Since we're reviewing equilibrium constants this week, let's talk a little bit more formally about what it means for the reaction to go to the right when we add more acid. So normally when the reaction is at equilibrium, that means it's been happening for a long time and there are as many reactants being converted into products as there are products converting back into reactants. There's a certain ratio between the concentrations for those different things, and that's determined by the equilibrium constant. So this is it turns out to just be 10 to the 4.5 for this particular reaction. So what happens right after we add more acid? The concentration of protons has gone up, and there's no time for the reaction to reach equilibrium again, so that means that this whole ratio will be less than the equilibrium constant. And in order for this reaction to get back to equilibrium, some of this stuff in the denominator is going to have to convert back to stuff in the numerator, and that's how we can tell that the reaction is going to go from the left over to the right. Another technique we commonly use in cooking to disrupt protein folding is just adding more salt to the food. So here again you have two amino acids involved in an ionic bond, and we're going to add table salt, sodium chloride, that's NaCl. And when the salt is added, it dissolves to make two oppositely charged ions, and those ions can substitute for the pairing that you had here so that you wind up with more ionic bonds, but they're not between amino acids anymore. And that will allow the amino acids to float apart from each other and lead to the protein unfolding. You've already seen one way that lowering the pH can disrupt protein folding, which is by neutralizing the charges on negatively charged amino acid side chains. But another way is that when we add these acids, they break down to produce free protons and also some negatively charged anion that varies for each acid. And those two different ions can also replace the ionic bonding that normally held the amino acids together. So why does unfolding proteins cause foods like eggs to become solid when we cook them? Well, imagine I have a bunch of proteins inside a cell, and so I'm going to draw their backbone as one long line like that, and the interactions that keep them folded as these ionic bonds between positive and negative charges. In reality, in a protein, hydrophobic interactions and hydrogen bonds would be contributing too. And what you're going to notice is that all of those bonds are intramolecular. They're between different parts of the protein, but they're not between the different copies of the protein. When we unfold the protein, we expose all of those different amino acid side chains that had been involved in intramolecular bonds, and that allows them to contact other proteins and form intermolecular bonds instead. And this holds the proteins together in a giant gel that gives foods like cooked eggs their solidity. So we'll talk about all of that a little bit more in the week on elasticity, But for now, that's the basic idea behind coagulation.